Okay, so before we do our microscope lab, let's go over a few things you need to know about using a microscope. So first of all, this microscope is a little bit different than what you might have had in middle school because it does have two ocular lenses instead of one. And then you can move those closer or further apart depending upon how far your eyes are from each other. So that's cool. Alright, so when you go to use the microscope and plug it in, and then back here in the back of the microscope, we've got on-off switch, a little rocker switch, and then in the front, there's this brightness control. So you can turn that, and as we turn it, we see the lamp right here getting brighter. Okay? All right, going up, right here where my hand is, this is two knobs, one embedded into the other. So the outside one is called the coarse wheel adjustment. And if you turn that, you can see that the stage goes up and down to get closer to your objectives. And then the little one in the middle of it, that's called the fine wheel adjustment. And it makes very, very small movements that don't, well, they're not noticeable from looking at the stage. But it does sharpen up your image there. Okay. And by the way, you do have these same knobs on both sides of the microscope, so if you're left-handed or right-handed, you can use it right. Okay, I'm going to scoop the microscope around a little because I want to show you something else, and that is this thing right here. So it's a little different than the stage clip that you might have had on your middle school microscope if you got a chance to use those. So what you're going to do is pull that little lever, and then you can fit your Then you can fit your slide in there, and it pushes so that your slide is up against the corner there, and it holds it in place. So that's, like I said, a little different than what you had in middle school. Another thing that is a little different from your middle school microscope are these stage controls right here. So there's one on top and one on bottom. And if you look at the slide while I turn this, can you tell that it's moving it forwards and backwards? And then the one underneath it, if I use that one, then it will move the slide left to right. So you don't physically touch the microscope slide after you have it up on the stage. You just use these two stage controls to center your specimen above that light source. Pretty cool. All right, next we have this thing here called the revolving nose piece, and it's got this grippy kind of feel to it, so that's where you hold on to it, and you just turn that to get from one objective to the other. The starting point is always going to be the red one, which if you can see the numbers on here, I don't know how well that can show up with the camera, but it says 4, and that means this magnifies 4 times. So in conjunction with the ocular lenses, which are 10, then your total magnification as viewed through here would be 40. 4 times 10 is 40. This is called your scanning objective, and like I said, that's where you're going to start. So you put your slide in there, you've got it on scanning, and then you look through your ocular lenses, and you're going to move the coarse wheel objective knob, which is a coarse wheel adjustment knob, which is the bigger one. You're going to move that while you're looking through the lenses until your object comes into focus. And once it comes into focus, guess what? You never touch it again. Never, never, never. Because from that point forward, you only have to make little adjustments, and that's with the little tiny little fine wheel adjustments. Okay? These microscopes are parfocal, which means that once you have focused your image on the scanning power, when you revolve this revolving nose piece to the yellow one, which is 10x, for a total of, did I see you do that? Going from 4x, you're going to revolve that around to the 10x. So that's a total of 100 times magnification. You don't move the coarse wheel at all. You already have it focused, so all you might have to do is adjust the fine wheel just a little bit down here. You don't use the coarse knob anymore, just that fine wheel to unsharpen up your image. You might, however, have to move the stage controls if you didn't have your specimen centered in the field of view. And the field of view is just the circle of light that you see when you're looking through your lenses. So again, if we're going to go up to the highest power, because the yellow is low power, the red is scanning, then the yellow is low. We're going to turn, and it looks like you're going to hit the slide, but you're not going to. And if you do, there's a little, like a spring mechanism in there that keeps it from damaging the slide or the lens itself. So you don't move that stage anymore because it's already focused, and you just use this little tiny focus. Okay, one other thing, there's one more objective lens that we won't be using, so I'm just going to tell you not to use it right now. This is the white lens, which is the oil immersion lens. You use this in conjunction with oil, and like I said, we're not going to be using it. So we're just doing the red, the yellow, and the blue, which is 4 times, 10 times, and then 40 times. So it goes up to 400 times magnification. The last thing that I want to show you on how to use the microscope is this little thing right here. This is called the iris diaphragm, and so you can turn this from side to side, and it's kind of like the iris of your eye, which are the muscles that control the size of your pupil. This is controlling the size of the hole that the light goes up through the stage opening. So it will make your image brighter or darker. And so you want to play around with that to see just what gives you the best image through it. All right, so then, real quick, how do you make a wet mount? All right, so if you look at our slide here, I've got a specimen that we're going to put on the microscope. So, because this is already sitting in water, I don't have to add any water to it. I just have to buff off a little leaf. This is LED. I'm going to put that on my slide. Actually, it's not as red as I would like. And then I'm going to take my cover slip, which I have over here, and I don't want to get fingerprints on it, so I'm just holding that on the edge. And I'm going to gently lower my cover slip down, and because of the properties of cohesion and adhesion, that cover slip is going to stick to the slide. Even if I turn it upside down, it's not going to fall off because of those properties. All right, so now, like we said before, we will put that up under our objectives, and then we do that whole process that I just talked about. Okay, so one of the other things that you're going to be doing is adding 15% saltwater solution to your slide, and see what happens there. So look carefully. You're going to put a few drops on the edge of the slide, right at the edge of the cover slip, and you want it to get pulled across underneath that cover slip. So you've got some paper towel squares here that is going to help pull that across. So you'll pull through. Can you tell that this is wet now? The paper towel square. So that pulled it through. So then you can compare what did the cells look like before the saltwater solution, and what do they look like after the saltwater solution. Okay, so hopefully you guys are good at how to use the microscope. When you're finished, you take the stuff off. Obviously, don't leave slides on the microscope. Make sure that your objective is back down to scanning power. Take this illuminator and turn it all the way down. Turn off the microscope. Unplug it. And then you'll put the cover back on it. Make sure your station is all clean and all that.